Good day, this is your Alan, and we are in the second quarter and the seventh concept to be discussed. We're going to discuss the muscular system, which includes the muscle physiology and also the muscle contraction. And these are our learning outcomes. At the end of this discussion, the students will be able to describe the general functions of the muscle, able to identify and describe the types of muscle, and describe the macroscopic structure of the skeletal muscle, describe the components of a sarcomere, describe an action potential, and also able to describe the events of muscle cell contraction and explain the sliding filament theory. There are three types of muscles. You have skeletal muscle, cardiac muscle, and also the smooth muscle. As we know that skeletal muscle are attached to the bones, and these muscles are striated and under the voluntary control. Where your cardiac muscle, it is located in the heart. It is also striated muscle and involves in involuntary control. Lastly, the smooth muscles, it is located in the blood vessels and hollow organs. This is non-striated and also involves in involuntary control. There are 7 functions of the muscular system. You have for movement, maintaining posture, for respiration, production of body heat, communication, heartbeat, and contraction of organs and vessels. There are 4 properties of muscle. You have your contractility, excitability, extensibility, and elasticity. When we talk about contractility, the ability of the muscles to shorten forcefully or to contract. While excitability, this is the capacity of the muscle to respond to any stimuli or stimulus. When we talk about extensibility, this is the ability of the muscles to stretch beyond its normal resting length and still be able to contract. And lastly, for elasticity, these muscles were able to recoil to its original resting length after it has been stretched. Skeletal muscle or striated muscle with its associated connective tissue constitutes approximately 40% of the body weight. The skeletal muscle is so named because many of the muscles are attached to the skeletal system. Some skeletal muscles attach to the skin or connective tissue sheaths. Other muscles or skeletal muscles are also called striated muscles because transverse bands or striations can be seen in the muscle under the microscope. Individual skeletal muscles such as the biceps brachii are complete organs as a result of being comprised of several tissues, muscle, nerve, and also connective tissue. Connective tissue coverings of a muscle includes epimysium, fascicles, perimysium, and endomysium. Each skeletal muscle is surrounded by a connective tissue sheath, and this is called as your epimysium. A skeletal muscle is subdivided into groups of muscle cells, and this is termed as your fascicles. Each fascicle is surrounded by a connective tissue covering, and this termed as your perimysium. Each skeletal muscle fiber is surrounded by a connective tissue covering, termed your endomysium. A muscle fiber is a single cylindrical cell with several nuclei located at its periphery. The muscle fiber range in length 1 cm to 30 cm and are generally 0.15 mm in diameter. The skeletal muscle fiber contains several nuclei that are located at the periphery of the fiber. The sarcolemma or cell membrane has many tube-like inward folds called transverse tubules or T-tubules. T-tubules occur at regular intervals along the muscle fiber and extends into the center of the muscle fiber. The T-tubules are associated with, in large portion, of smooth endoplasmic reticulum which is called as your cytoplasmic reticulum. The enlarged portion of are called terminal cisternae, and the tubule connects the sarcolemma to the terminal cisternae to form a muscle triad. The sarcoplasmic reticulum has a relatively high concentration of calcium ion, which plays a major role in muscle contraction. The cytoplasm of a muscle fiber is called the sarcoplasm, which contain many bundles of protein filaments. Bundle of protein filaments are called myofibrils, and these myofibrils consist of myofilaments, the actin, and the myosin. Actin myofilament or thin filament is one of the protein fibers that make up a sarcomere. So in this picture, this is the actin myofilament. The thin filaments resembles two-minute strand of pearls, while your myosin, and this is your myosin, myofilament, okay? So, or a thick filament, it is another protein fibers that make up a sarcomere. So the thick filament resembles a bundle of golf clubs. Your myofibril, okay? So this is your myofibril, okay? It's a fine longitudinal fibril within a skeletal muscle fiber. And also your eye band, okay? So this is your eye band, is the region of a striated muscle sarcomere that contains the thin filaments. And your A band, so this is your A band, okay? So it's a region of a striated muscle sarcomere that contains myosin thick filaments. And your H zone, so this is the H zone, is the center of A band where there is no overlap between the thick and the thick and thin myofilaments. Your M line, so in the middle, this is the M line, okay, is the attachment site for the thick filaments. It is the center of A band, and it is the center also of the sarcomere. Your C disc, so this is your C disc, okay, defines the boundaries of a muscle sarcomere. Two adjacent Z disc along with this myofibril mark the boundaries of a single sarcomere. So another component is your troponin. It is a complex of 3 regulatory protein that are integral to muscular contraction in skeletal and cardiac muscle, but not on the smooth muscle. And your tropomyosin, it is a 2-stranded alpha helical coiled coil protein found in the cell cytoskeleton. Now we are going to discuss on the skeletal muscle fiber functions or action. Starting from a sarcomere, it is the basic structural and functional unit of a skeletal muscle because it is the smallest portion of a skeletal muscle capable of contracting. The C disc form a network of protein fibers that both serves as an anchor of an actin myofilament and separate one sarcomere from the next. A sarcomere extends from one Z disc to the next Z disc, and this organization of actin and myosin myofilament gives skeletal muscle its striated appearance and gives it the ability to contract. The myofilament slides past each other, causing the sarcomere to shorten. With each sarcomere consists of 2 light staining bands separated by a dark staining bands. And these light staining bands consist only of actin and are called I band that extends toward the center of the sarcomere to the end of the myosin myofilaments. The dark staining bands are called A band that extend the length of the myosin myofilament. Actin and myosin myofilament overlaps from some distance on both ends of the A band, and this overlapping causes the contraction or muscular contraction. Actin myofilaments are made up of three components: actin, troponin, and tropomyosin. Troponin molecule have binding sites for calcium, and tropomyosin filaments block the myosin myofilament binding sites on the actin myofilament. Myosin myofilament or thick myofilaments resembles the bundles of a tiny golf clubs, and the myosin heads are ATP binding sites, ATPs, and attachment spots for the actin. Now we are going to talk about excitability of muscle fibers. The electrical charge difference across the cell membrane of an unstimulated cell is called the resting membrane potential. Muscle cell or fibers have a resting membrane potential but can also perform action potential. The resting membrane potential is due to the inside of the membrane being negatively charged in comparison to the outside of the membrane being positively charged. The action potentials are due to the membrane having a gated channels. To initiate a muscle contraction, the resting membrane potential must be changed to an action potential. Changes in the resting membrane potential occur when gated cell membrane channels open. In a skeletal muscle fiber, a nerve impulse triggers gated sodium channels to open, and sodium diffuses into the cell down to its concentration gradient and toward the negative charges into the cell. The entry of sodium causes the inside of the cell membrane to become more positive than when the cell is at the resting membrane potential. This increases in positive charge inside the cell membrane, and this is called as your depolarization. If the depolarization changes the membrane potential to the value called threshold, an action potential is triggered. An action potential is a rapid change in charge across the cell membrane. Depolarization during the action potential is when the inside of the cell membrane becomes more positively charged than the outside of the cell membrane. Near the end of the depolarization, the positive charge causes the gated sodium channels to close and gated potassium channels to open. Opening of the gated potassium channel starts the repolarization of the cell membrane. Repolarization, this is due to the exit of potassium from the cell, and outward diffusion of potassium returns to the cell to its resting membrane potential condition. And the action potential ends. In a muscle fiber, an action potential results in a muscle contraction. Now moving on, a motor neuron is a nerve cell simulates muscle cell, while your neuromuscular junction is a synapse where the fiber of a nerve connects to a muscle fiber. A synapse refers to a cell-to-cell junction between a nerve cell and either another nerve cell or an effector cell, such as in a muscle or a gland. A motor unit is a group of muscle fibers that a motor neuron stimulates. A presynaptic terminal is the end of the neuron cell axon fiber. The synaptic cleft is the space between the presynaptic terminal and postsynaptic membrane. And the postsynaptic membrane is the muscle fiber membrane called your sarcolemma. A synaptic vesicle is a vesicle in the presynaptic terminal that stores and releases neurotransmitter chemicals. Neurotransmitter are chemicals that stimulate or inhibit the postsynaptic cells. Acetylcholine is also a neurotransmitter that stimulates skeletal muscles. Function of the neuromuscular junction. An action potential arrives at the presynaptic terminal, causing calcium ion channel to open. Calcium ions enter into the presynaptic terminal and initiate the release of a neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. So the diffusion of acetylcholine across the synaptic cleft Unbinding the acetylcholine receptor sites on the postsynaptic muscle fiber membrane opens the sodium channels. Sodium ions diffuse their concentration gradient, which result in depolarization of the muscle fiber membrane. If threshold is held, or if threshold has been reached, a postsynaptic action potential is achieved. We are going to talk about the muscle excitation. An action potential travels along an axon membrane to the neuromuscular junction. Calcium channels then open, and calcium enters into the presynaptic left terminal. Acetylcholine is released from the presynaptic vesicles with the aid of your calcium ion. As a result, acetylcholine stimulates these sodium channels on the postsynaptic membrane to open. As a result, sodium diffuses into the muscle fiber, initiating action potential that travels along the sarcolemma and the T-tubule. Action potentials in the T-tubules causes the sarcoplasmic reticulum to release calcium ions. Action potentials in the T-tubules causes the sarcoplasmic reticulum to release calcium. On the actin calcium bind to troponin, which moves the tropomyosin and exposes the myosin attachment sites. ATP molecules are broken down to ADP and P or phosphate, which release energy needed to move the myosin heads. Then the heads of the myosin myofilaments bend, causing the actin to slide past to the myosin. As long as calcium is present, the cycle repeats. Now we are going to talk about ATP breakdown and cross-bridge movement. So energy for muscle contraction is supplied by ATP. Energy is released as ATP will be breakdown into ADP plus phosphate. ATP is stored in the myosin heads, and ATP help from cross-bridge formation between myosin and actin. New ATP must bind to myosin before cross-bridge is released. Before cross-bridge cycle, calcium binds to the troponin and tropomyosin moves, exposing active sites on actin myofilament. The myosin heads bind to the exposed active sites on the actin myofilaments to form cross-bridge, and phosphates are released from the myosin head. Energy stored in the myosin head is used to move the myosin heads, causing actin myofilament to slide past the myosin filaments, and ADP molecules are released from the myosin heads. An ATP bundle or an ATP molecule binds to each of the myosin heads, causing them to detach to the actin myofilament. Now, the myosin ATPase portion of the myosin or the myosin heads split ATP into ADP phosphate, which remain attached to the myosin head. And lastly, what will happen now is the head of the myosin molecules returns to the resting position, and energy is stored in the head of the myosin molecule. If calcium is still attached to troponin, so cross-bridge formation and movement are repeated. This cycle occurs many times during the muscle contraction. Not only cross-bridge form and also release simultaneously. Energy for muscle contraction. Muscle fibers are very energy demanding cells, whether at rest or during any form of exercise. Energy comes from either aerobic with oxygen or anaerobic without oxygen ATP production. ATP is derived from 4 processes in skeletal muscle. Okay. Aerobic production is found in the cytoplasm and mitochondria through Krebs cycles, oxidative phosphorylation, and glycolysis that produces at least 36 ATP. During anaerobic production, it comes from the cytoplasm through glycolysis and fermentation that produces only 2 ATP. How about your creatinine phosphate? Your creatinine phosphate functions is in the muscle cells by storing energy that will be transferred to ADP to resynthesize ADP. So now on the fourth, which is your conversion of ADP to 1 ATP and 1 AMP or adenosine monophosphate, it is done during the heavy exercises. Muscle twitch is a single contraction of a muscle fiber in response to a stimulus. So if this is the stimulus, and this is the contraction phase, and this is the relaxation phase. A muscle twitch has 3 phases: latent phase, contraction phase, and relaxation phase. The latent phase is the time between the application of stimulus and the beginning of a muscle contraction. The contraction phase is the time during which the muscle contracts, and the relaxation phase is the time during which the muscle relaxes. We are going to compare the muscle fiber types. You have your slow twitch fibers and the fast twitch fibers. For the slow twitch fibers, it contracts slowly and fatigues slowly, have a considerable amount of myoglobulin, and it uses aerobic respiration. The color of the muscle fiber is darker and it is used by a long distance runners. However, the fast twitch fibers contract quickly, fatigue quickly, and it is used anaerobic respiration. Energy is from glycogen, and it is lighter in color, and it is used for the sprinters. A muscle has a blend of types with one type dominating. Humans have both types of fibers, and the distribution of fibers are genetically determined. What is muscle fatigue? Fatigue is a temporary state of reduced work capacity. Without fatigue, muscle fibers would be worked to the point of structural damage to them and their supportive structures. So what are the mechanisms of muscle fatigue? This includes acidosis and ATP depletion due to either an increased ATP consumption or a decreased ATP production. Oxidative stress, which is characterized by a building up of excessive reactive oxygen species or ROS. These are free radicals. And lastly, your inflammatory or your local inflammatory reactions. So before identifying the different types of muscles, its location, its origin insertion, and its action, we need to identify first or know the different types of muscle contractions. So there are 2 types of muscle contractions. You have your isometric and you have also your isotonic. Isometric contraction has an increase in muscle tension but no change in length. Your isotonic contraction has a change in muscle length with no change in muscle tension. Concentric contractions are isotonic contractions in which muscle tension increases as the muscle shortens. Your eccentric contractions are isotonic contractions in which tension is maintained in the muscle, but the opposing resistance causing the muscle to lengthen. So that's it. So this is the end of our discussion or topic for our lecture. So thank you so much and have a blessed day.